Darwin wrote his The Origin of Species, all about synthesis and evolution. Scientists cannot say how that evolution happened. This caused other scientists to get involved and resulted in modern synthesis, which organized the basic principles that have guided evolutionary biology today. Darwin insisted that embryology was crucial to the understanding of evolution. He said that it was, quote, the strongest single class. There was a conflict with this idea. It was hard to explain how different organisms could come from eggs that appeared to be the same. Another challenge was that until recently, nobody knew which gene differentiated between animals. For example, nobody knew how one gene could cause a difference between a fly's wing and a bird's wing. The fruit fly became the test rat, per se, to bridge this gap. The toolkit of genes, the basic bodybuilding genes that organisms share, was discovered as a result of this. These toolkit genes can explain how different animals have similar structures. Advances in the new science of evolutionary developmental biology, dubbed EVO-DEVO for short, have enabled biologists to see beyond the external beauty of organic forms, mechanisms that shape their diversity. Many genes that coded for things in a fly had similar counterparts in a human. This shows that there's the, like I said, toolkit of bodybuilding genes. EVO-DEVO teaches us about diversity. Originally, it was thought flies and fish, for example, must have vastly different genes. They are different animals, after all. They did not account for the possibility that there were similar sets of genes to make different structures. For example, eyes all share a similar gene called the PAX6 gene. This PAX6 gene has been seen to command everything from compound eyes, single lens eyes, photoreceptors, and light-sensing proteins. This proves how eyes didn't just pop into existence in the case of different species. They're a variation of one existing gene. Studies have shown that the variance in how an organism uses the genetic toolkit contributes to its uniqueness. For example, an obvious feature of many large animals is the construction from repeating parts. Arthropods have segmented bodies, vertebrates have segmented vertebrae. Subsets emerge from these building blocks, like appendages or ribs, respectively. Another variance is the change in the kind and number of these repeating parts. There are different vertebrae and number of them in an organism's backbone. Number of segments and kind of appendage likewise varies in arthropods. Number and structure are formed by a type of gene known as a Hox gene. Genes govern identity and the number of some structure, and likewise a large body of variance shows that Hox genes are shifted from being expressed to not expressed between species. Shifts explain how humans and snakes are alike. Both of them have vertebrae, but shifts in Hox genes cause different number of vertebrae to be in humans or snakes. Other structures are the same. Hox gene expression shifts can account for both large-scale and interspecies variance. For example, the three-spined stickleback fish has two forms. While both are the same species, one form is a bottom feeder with small spines. The other form swims freely with big spines. The small spines allow the bottom feeder to not be attached upon by dragonfly larvae and eaten. Big spines, however, allow the free swimmer to not be swallowed by larger predators. These are both caused by Hox gene shifts and natural selection. Whatever is better for survival of an organism is therefore kept. These Hox gene shifts occur in obviously the same species, but this can mean that they have different structures.
Evolution can change the number and kind of repeated body structures. Bird feathers, for instance, are prominent examples of the novelties that have emerged from the changes in the ways the toolkit genes are expressed. Humans have long supposed that we hold some unique position in the animal kingdom. In reality, as molecular biologists have now know from sequencing the genomes of our own and other species, is that the genes of human beings are very similar to the genes of chimpanzees or that of the mouse. In fact, all other vertebrates. A more likely explanation will come from understanding how our old genes shared with other primates, mammals, vertebrates, and more distant animals relate. Darwin knew very well the difficulty people would have in picturing how complex structures arose. Darwin said, "Contrivance must have a contriver, design a designer. It is only the display of contrivance that the existence, the agency, the wisdom of the deity could be testified in his rational creatures." Paley's argument to the existence of the idea of intelligent design is now being touted as a new alternative to evolutionary science. Evo Devo shows how complex forms and structures evolve, not only in the ways that lead from one species to the next, but also in the ways such as making of body plans that have shaped the major differences in higher taxonomic ranks. The major tenet of the modern evolutionary synthesis is the evolution forms above the species that can be extrapolated from processes operating at the level of populations within the species. Through the lens of evolutionary development, biology and biologists can finally see beyond external forms in the very process that forges them, completing the picture of how the endless forms of nature have been and are being evolved.
Let's summarize. Here are 10 key things you need to know. Darwin's original vision of evolution and the origin of life makes the basis of all biological research and studies that have been done over the past 50 years. Essentially, he thought that all complex organisms originated from simple organisms. It is believed that development is the reason that animals become the way they are. They had no idea that different forms could actually be built with similar sets of genes, but then they found out that they shared some common genes when Walter Geering discovered that a gene required for eye formation in fruit flies is the exact counterpart of a gene required for eye formation in humans and mice. The great diversity in crazy quilt distribution of eyes throughout the animal kingdom was, for more than a century, thought to be a result of the independent invention of eyes in various animal groups. Discoveries in Evo Devo have forced the thought of re-examination of eyes. Advances in new science of evolutionary developmental biology, dubbed Evo Devo for short, have enabled biologists to see beyond the external beauty of organic forms into the mechanisms that shape their diversity. The new knowledge of toolkit genes make it clear how complex structures are built, and Evo Devo makes it possible to connect this process to long-term processes of evolutionary change. Paley's argument in the essence of the idea of intelligent design now being touted as a new alternative of evolutionary science. The major tenet of modern evolutionary synthesis is that the evolution of forms above the species level can be extrapolated from processes operating at the level of populations within species. Here's a crossword puzzle to test your understanding of what we just told you. Pause the video now and complete it.